Hello, hello. Good morning, good afternoon, good evening, depending on where you are. We'll give folks a couple of minutes to join in here and trickle in. Go ahead and add in the chat where you're tuning in from. We typically have folks from all over the world. When you do use the chat, if you want to chat to everyone, make sure to hit that little drop down that says everyone. Otherwise, you're just talking to myself and Tiffany. My name is Cody Hanson. I'm on the team at Singularity. Very excited to be bringing you this monthly discussion series where we dive into the world of exponentials, and very excited to have Dr. Tiffany J. Vora, with us today. And we're going to be talking about synthetic biology. Behind the scenes, we have Alex Rupsam, who is our Director of Expertise and Knowledge, and she'll be moderating the chat and helping us out with any links and things. But that is the team that you have for today. And I think we're at a good number here, so let's go ahead and get started. So I've introduced myself. Tiffany, I think I'd love to pass it over to you to introduce yourself. I think you can do a better job than I can. So let's get started there.
Hi, everyone. I'm Tiffany Vora. I'm coming to you today from Connecticut, so on the East Coast of the United States. I'm a molecular biologist by training. I did my PhD in molecular biology, what seems like a really long time ago now, just at the beginning of systems biology and computational biology. I've worked in industry and academia. I also used to be a full-time faculty member at Singularity University, as well as our faculty director. So I have a long, wonderful history with the S universe, and I'm delighted to be back today to have this conversation with you about synthetic biology. So with my background in molecular biology, I'm interested in biotech and everything that it touches. I think it's a really great time to be a biologist because biotech now is becoming so powerful for reasons that I'm sure we'll talk about, that whether you're interested in agriculture, healthcare, energy, materials, space, just about anything, biotech might have a way to be turbocharging your innovation and your business. So, like I said, best time ever to be a biologist, and so excited to have this conversation today.
Awesome. Thanks, Tiffany. Let's jump right in kind of just from the beginning. So I'm a beginner to the biotech space. I can assume I think a lot of folks tuning in maybe have varying levels of background and expertise, but I think it's something that you live every day and most of us don't. So I'm very excited to get your perspective on the world from this lens. So just starting at a very high level, what is synthetic biology? I know you also speak on digital biology. That's also a term
Mhm,
people hear a lot. So what's the difference,
mhm.
and what's going on in that world?
Sure. So let's start with digital biology, because I think that's a really nice mental framework for then going into any of the detailed sub areas of biology. Synthetic biology, I would say, is one of those. So, digital biology is this framework that we talk about a lot at Singularity University. Raymond Macaulay was one of the folks who kicked off the use of this term. Digital biology is thinking of the information of life the way that we think about information on a computer. So, computers store information as zeros and ones. On Earth, life stores information as DNA. So, as the building blocks A, C, T, and G, which are short for some chemical names that we use. So, that's a really useful mental model because if you can think about life storing information as A, C, T, and G, then essentially anything that we can do with computer code, we can also do with DNA code or other types of biological code. So, if we move big pieces of DNA code around, that's called genetic engineering. We've known how to do that since the 1970s. If we write DNA code from scratch, that's synthetic biology. We're going to talk a bit about that today. And then, of course, if you go in and debug the source code of life one letter at a time, that's called gene editing. And there's all kinds of great technologies like CRISPR and base editing that empower you to do that. So that's the big mental model. And I mentioned synthetic biology is one of the areas that we can use that mental model as beginners to start accessing. So, what's synthetic biology? Synthetic biology is when we combine biology, engineering, and computer science in order to design and construct and modify biological systems for specific purposes, contexts, and applications. So, essentially, the goal is to take the biological systems that we already have, thanks to about 4 billion years of evolution on this planet, and then use the tools that we're developing today in order to either have better performance or even have entirely new functions that, to our knowledge, nature hasn't used here on Earth. So why do we want to do this? And it's because biology is amazing, right? Biology is self-perpetuating. Biology is modular, it's flexible, it's programmable, it's resilient. And as long as you keep DNA cold and dry, it can be stable for decades to millennia to more than millennia, hundreds of millions of years even. So there's just this really awesome potential for synthetic biology to be used across innovation spaces. And actually, that gives us a chance to hit on a point we wanted to make sure that we mentioned today, which is that synthetic biology is not itself a vertical. Instead, I invite you to think of synthetic biology as an enabling technology that underlies lots and lots and lots of other verticals. So you can use SynBio for agriculture. You can use SynBio for environmental remediation. You can use it for food. You can use it for healthcare. You can use it for energy. You can use it for materials. All of those things have the potential to be harnessing this synthetic biology revolution that we're in today.
That's a great point, and I think that in a lot of cases, digital biology, synthetic biology are not being given its fair share of voice compared to just digital technologies like AI, etc. And it seems from an outsider's perspective like there's an incredible amount of potential that remains untapped, um, possibly simply because of quote unquote like an exposure challenge or a marketing challenge among um, people in corporations, people in venture capital, et cetera, that are maybe just not fully aware of the potential. And so I think that's something that I would hope that we can share more of today, and that we'll share more of at the Future of Biotech program that we're discussing later on as well, is tapping into this potential and making it bringing it to light for folks that maybe haven't been exposed to it in the past. With that in mind, what are some recent breakthroughs that you're just excited about, like, news headlines? And quickly before you answer, I want to encourage anyone to use the Q&A function in the Zoom. We're going to dive into Q&A with Tiffany here at the last 15, 20 minutes, depending on how many questions we have. So as you think of questions, please add those into the Q&A tool at the bottom, and we'll get to those toward the end of the hour. But thanks, Tiffany.
Yeah, yeah. So, gosh, so much I could have responded to and what you just said. Before I answer your question about breakthroughs, I do want to point out that one of the things that's really interesting about working in biology versus, say, working in digital apps or something like that is that the biological world is this fully formed system on its own, right? So biology is hard because there is this underlying objective reality out in the universe and biological principles that are at play. And we have to work those out as we figure out how to use them. Whereas when you're working with an app, that's a completely man-made system, right? So the thing that makes synthetic biology hard and interesting and fascinating and weird and surprising is this overlap between the human and the natural worlds, and now the silicon-based world, to get all that going. So I think it's not just a matter of people don't know about SynBio. It's hard. It's rewarding, and there's lots of potential there. But it's not just programming a widget to pop up on the App Store. There's this other thing there. So, like I said, that's what makes it really interesting.
It's a great point.
Yeah. So you asked me about breakthroughs in synthetic biology. So I'll give you kind of two easy ones, foundational ones, and then I'll give you two slightly more technical ones. Does that sound okay?
Sounds great.
So cool. So I think the first breakthrough that has happened and is continuing to happen is just how cheap, fast, and accurate it is to carry out the basic functions of synthetic biology. So the first one of those is DNA sequencing. Sequencing, when we talk about these technologies, is just the ability to read out that ACTG order. And if you can remember 20 years ago, 20, 25 years ago in 2001, when the first draft of the human genome was co-published at the same time by a public team and a private team, it had cost a small number of billions of US dollars in order to sequence that first human genome. And today you can sequence a genome for on the order of 500 US dollars. So the price went from being as much as a fighter jet to being as much as a ticket to a Taylor Swift concert, right? So that's a really big thing. And that's to do the whole sequence. If you just do a couple pieces of the human genome, then you can be talking about like $200, which is your Spotify account for the year, right? And what makes that so empowering for synthetic biology is that that means it's cheaper to not only sequence human DNA, but to sequence the DNA of everything. And the DNA of everything is the raw material for synthetic biology innovation. So that's really important. The second foundational piece that's gotten cheaper, faster, more accurate, more powerful is DNA synthesis. So if sequencing is reading, synthesis is writing. It's not on quite as an aggressive price performance curve as DNA sequencing is, again, for reasons that have to do with chemistry and computation. But DNA synthesis is getting cheaper in accordance with something like Moore's law. So that's great. We're also able now to print out longer pieces of DNA, which is important because one of the things you have to do in synthetic biology is put pieces of DNA together. So the longer and more error-free the pieces of DNA you're working with, the fewer assembly steps you have to do, which means overall it's just easier to do the thing. So as I'm watching this space, I'm watching companies that are getting cheaper, faster, and more accurate at making these longer pieces of DNA that they can stick together. So again, reading and writing, two fundamental technology bases for synthetic biology. Um, I'll tell you really briefly, another breakthrough that I think is really interesting as a scientist is how much powerful CRISPR has gotten. So why do we need CRISPR for synthetic biology? So as a reminder, CRISPR is a system that is based on the bacterial immune system. So it was not invented by humans. That allows you to go in and precisely and permanently change the DNA code at the ACTG level. And again, base editing is a similar idea. You can think of base editing as using the eraser on a pencil instead of a pair of scissors like CRISPR does. And from a fundamental science perspective, the reason this is so important is because it gives us the power to rapidly screen incredibly large parameter spaces of biological molecules. So again, it's a way of getting at the inputs that would go into a SynBio system and rapidly use them for things we want to do with them. You put that together with AI, which, Cody, I'm sure we are going to talk about AI and biology, and now suddenly not only can you go cheaper, faster, more accurately through the DNA, the molecular parameter space that Earth has already been experimenting with through evolution, but now you can start looking at things that, to our knowledge, nature has never tried out on Earth before to come up with entirely new things. And man, I mean, I don't know if you can see it on my face, you guys, but like this is the most boring biology is ever going to be moving forward. And I think it was never boring. So for me, this is fantastic. It's only going to get better from here.
Yeah, your playground is like just being built, and now you can run from here. Yeah, I was gonna go into AI next because I think that's on everyone's minds, and obviously we have LLMs out there. You can write, whatever poems and create cool images, but um, there's a lot being done in the combination of using AI in the biotech space, and I mean, the groundwork that you just laid there of being able to read, write, and edit DNA as something of a language, a coding language, and then combining that with the power of AI to write and create coding languages. I think it's almost like there's too much to imagine for a layperson. So, like, what do you see happening with biotech and AI, and obviously like AlphaFold was one of like the big, announcements over the past decade of using AI to read how proteins are created. That's maybe a starting point. But where do you see this going with AI and the ability to write or program life?
Yeah, so many places. So first, I want to remind us that we had AI before ChatGPT and before generative AI and before LLMs. So I'm going to take you back a little bit and then bring us forward to that point. So, when we talked about natural language processing, machine learning, computer vision, all of these things, they were being applied in really interesting ways in biology and the various verticals that biotech touches, like healthcare, way before this current AI moment that we already have. And it's kind of funny when I remember. So when I was in graduate school, gosh, 25 years ago now, which really like isn't that long ago, all things considered, I was inventing a technology in the laboratory that I thought, figured, great, this is great. We need this tool. And then suddenly I got it to work. And then I had 19 million data points and I had no idea what to do with them. And so suddenly I had to learn to program in order to handle all that data. And back then, being a biologist, most of us sort of just had to be able to multiply, and that was considered sufficient. And then suddenly I was in this field where you had to have advanced mathematics, statistics, all kinds of advanced math, and then be able to program on top of that. Fast forward now, and I think graduate students are understanding that actually computation is not a nice to have in biology. It's almost necessary. And especially if you're doing anything with large data sets, you've got to be fluent in this. So you mentioned AlphaFold. So now we'll fast forward to this point. So in my opinion, pirate poetry, which yeah, I use ChatGPT for all the time. That's not the killer app for AI. For me, biology is the killer app. So you mentioned AlphaFold. So as a reminder, AlphaFold was an algorithm that was released by Google DeepMind in the UK. And essentially what AlphaFold does is the first version of AlphaFold was it was able to predict a protein's structure from its sequence alone. I said that in one sentence, but that was the holy grail of molecular and cellular biology for a century. And again, when I was in graduate school, in order to solve a protein structure, you would use X-ray crystallography and do all these things. And it might take you 8 years, it might take you 10 years to solve a protein structure. You still have to do crystallography and things like that today, but now with AlphaFold, you could do that. They have a database now of every single protein known to man, right? And that's hundreds and hundreds of millions of them. It's there. These are predictions. You still have to go to the lab and confirm that the prediction works, but these predictions are pretty darn good. And so that means you can get 85% of the way, 90% of the way in 150 milliseconds instead of 8 years, which is pretty spectacular. I remember the word had gotten out in the community just before this algorithm dropped, and my husband's a professor at Stanford, and he's a biologist, a cellular biophysicist, and I said to him at dinner, "Are your grad students afraid that this algorithm is going to take their jobs?" And he said, "Are you kidding? They've already got the server set up, so the second the code drops, we can start running this. They don't want to do this anymore. They want to start from this point and then drive the science forward from there." And I'm like, awesome, right? Amazing and enabling technology. So now AlphaFold can do the shape of every biomolecule, not just proteins, but RNA and carbohydrates and other types of things. It can also now do the way those molecules come together, which is the fundamentals for, say, how a drug works with a receptor on something, which is amazing. And now this algorithm, as well as some other algorithms out there, are doing things like predicting new proteins that have new functions. So that's the part where I get really excited because AI now means that we can start looking to build in functionalities using biological systems that, as far as we know, biology has never done that before. And so there's just so many potential applications for this. It's really awesome. You mentioned LLMs, so let me just point out as well that when we look to apply AI to biology, biology is a series of languages. It's a series of nested languages. They're just chemical languages, right? So now what we're seeing are folks who are using LLM type approaches to do things like build antibodies to target particular diseases like HIV or Ebola or influenza A, and to do that much faster than we've ever been able to do before. We're seeing people using this for biological materials generation so that you can very quickly move to a set of candidates that you can prioritize to try out in the lab. And I guess we can't be all sugar and rainbows. I will also point out that folks are very concerned about the confluence between bioweapons and AI or chemical weapons or AI, because everything that you can do cheaper, faster in using AI for good purposes, there is potentially the odds that you could do that for bad purposes as well. Obviously, folks are very concerned about this, and there's a lot of work being done on this around the world, some of which I'm involved in, which I'm grateful for. But what that means, though, is that the choices that we make for these technologies, this is the moment where our choices really matter. And I find that a really empowering place to be.
Yeah, that's amazing. And I think a really good case study of the example you gave of your husband at Stanford of how people should be looking at AI in general, right? Of it's not taking our jobs. It's giving us the opportunity to go solve these new problems that we just didn't have time to go be creative and solve in the past. And I think there's no shortage of potential for what humanity can do with given the mindset and then given, empowered by AI. But moving on from that, I want to talk about application spaces. I think a lot of people relate biotech in general and digital biology and synthetic biology to healthcare just because it's an easy, connection for someone who, doesn't have kind of the education or exposure in this space. What are some application areas outside of the common ones, the ones you might expect that are very interesting right now? And I know, that you're doing some interesting work across some of these as well yourself.
Yeah, so let's look, for example, at agriculture. So folks are using synthetic biology and other types of biotechnology to do things like make crops that are more climate resilient, that can be more drought tolerant, heat tolerant, salt tolerant, any of these things. That's really important. I'm really excited about the folks who are using these technologies to do things like plastic remediation. So it turns out there are enzymes that biology has already innovated on through evolution that can break down plastics. And so folks now are using these tools to try to make those enzymes more efficient, more powerful, and to move them into other types of living things. So let's say, for example, you had a soil probiotic and you wanted to be able to break down some plastic waste in the soil. Folks are looking at how we can use these synthetic biology and genetic engineering techniques in order to make that actually functional. One of the things that I like about that is a concern that people have, which I understand, is that, well, what happens if you let these things out in the wild? Could they damage the ecosystem? Well, if you're trying to use synthetic biology to get rid of plastic waste in the ground or in the ocean or in our drinking water, what we're doing is we're using the technology to help an ecosystem that we've already damaged. And I think that's a really empowering way to look at this. So lots and lots and lots of agriculture space here, and some of this you've probably already seen. If you ever seen the black tomatoes that have all the lycopene in them, like that's a really nice example. So there's lots of these pieces that you can look at. So that's agriculture. Materials is a really interesting space. So there's a great company that I'm following called Lanzatech, where what they do is they have this system that they bolt it onto the manufacturing system that you've already got, and the microbes in there grab the carbon that your company is already emitting, and then they use that carbon to make things like yoga pants and sustainable jet fuel and all of these other things using that carbon as an input to a system. So that unlocks alternate revenue streams in a really exciting way to build all these other things, and also is helping with the carbon emissions, and you can think of it as a part of a circular economy. So from a really, if we zoom out one more step, I would say circular economy is another lens that we should be thinking about synthetic biology through, because biology is so flexible about taking inputs. So that's the materials end. And then I'll just say really quickly, another industry that I'm really excited about for synthetic biology is the space industry. So I also do a lot of work in space. My nonprofit Explore Mars, last year we sponsored a working group that I was also a part of to do a manuscript that we released that was about thinking about why we should be using synthetic biology for space, particularly for human missions that might be long duration. Say, for example, if you're going to go to Mars for a few years. And some of the big affordances to that are, again, this circular model. So you don't want to take anything with you in space that's going to be waste. Wouldn't it be great is if you could use that for input to make medicine or to make food or to make building materials or things like that? And also, it enables you to go from a take mentality, like bring it with you, to a make mentality, which helps with your up mass constraints and also means that you can have fresh stuff, fresh food, fresh medicine, instead of stuff that's been sitting around for 2 or 5 or 10 years. So, you asked me like what other industries do I see being transformed by synthetic biology? My answer is potentially every single one. And I, again, find that really exciting. Of course, I'm a biologist. I'm biased in that way, but I think we see lots of action across industries looking to see where can synthetic biology really be delivering value all through the value chain.
Are you are you imagining more of like an Avatar world versus like a Terminator world here in the future?
Um, I mean, I
I hope
have to
we all
believe
are.
in yeah, I have to believe in an Avatar world. If I believe in a Terminator world, then what are we doing, right? It doesn't
Exactly.
doesn't even make any sense. So yes, I'm pro, well, maybe less of the burning of the trees, but
Right.
I'll give you the rest of it. Yeah.
Okay. All right. We have more questions prepared, but we're getting a ton of questions in the chat. So I want to make sure that we have time to dive into as
Sure.
many as possible. So, Alex is behind the scenes kind of organizing some of these questions into themes. And
Okay.
so let's just start diving in, and then if we have time at the end, we'll get back into some of the questions that I had. But this is a great problem to have. So, the first are a couple from one's from Bruce, one's from Stu, about the practical applications of DNA synthesis. The first one is if you create a new organism by sequencing and synthesizing the genome, how do you set the epigenetic overlays that will help set the operation of the organism? And then the other kind of follow on to this is just what are some of the practical applications of DNA synthesis?
Sure. So let's start with the epigenetics part first. So, Bruce, thank you very much for that question. So, as a reminder, epigenetics, the word epigenetic means epi to stand upon, and genetic meaning the gene or the DNA molecule itself. So, in addition to the level of information that is at the ACTG level, there's now another layer of information that is contained by chemical marks that lie on top of the ATGG, like physically. I'm speaking about a physical reality here. And you can think of that a bit like highlighter in a textbook, if it helps you think of it that way. And so one of the big signals that these epigenetic marks send is an on or an off signal, which has to do with how tightly the DNA is wound. And again, these epigenetic marks change whether the DNA is open or whether it's coiled up. And when it's open, it's on, and when it's coiled up, it's closed. So the short answer for this is there is definitely work that has been done with looking to see how you can overlay the epigenetic code onto synthetic biologies, like synthetic DNA molecules. But the truth of the matter is, and certainly when I was doing this work with my own hands, we didn't worry about it this much because the biology kind of sorted itself out. Now, if you were trying to do this in humans, I would say, eukaryotic systems like human systems have much more reliance on these epigenetic mechanisms in order to properly turn on, turn off, or dose regulate, kind of tune how much expression you're getting. But with yeast and with bacteria, actually, humans don't have to intervene that much. The epigenetics sorts itself out. This is kind of another wonderful thing about working with biology is biology sort of figures it out. The human doesn't have to figure out every single one of the engineering principles. That's at a first pass. I am sure there are biologists out there who have done some really awesome work with specific tuning of epigenetics, but you don't hear a lot of talk about that with the microbes. One of the things we didn't talk about when we were talking about breakthroughs had to do with computational modeling. We now have far more powerful computational models of what biology is doing at not just the ACTG level, but the epigenetic level. And folks have been looking to see, what do we need to know about what those marks are? Do I need to have a lysine here, a methyl group here, and to be able to more powerfully get those tuned out. But, for a lot of it, the biology takes care of itself. The second question was about practical applications.
Yeah, of DNA synthesis.
Yeah, so gosh, anytime you want to make something, okay, I'll give you two. So one of them, first of all, has to do with the actual input-output system. So synthetic biology thinks of DNA as the software code and the living thing itself as the hardware that boots up the software, right? So that, for example, is how the mRNA vaccines for COVID worked. You had an RNA, which at this point, if you want to think of DNA as code, you can think of RNA as compiled code. That was encapsulated and then injected into your arm. And then your body was the hardware that booted up that software. So those RNA molecules had to be made before they could go into the vaccine and then come into your arm. So when we are doing things with DNA, DNA is a physical substance and you have to make it before you can do something with it. So whether that's making a vaccine, whether that's doing genetic engineering, whether that's making a biomedicine, whether that's engineering your tomatoes to make more of a particular vitamin, you've got to actually have the fragment of DNA before you can use it. Then once you pop it into the biological system, then the biological system will just copy it over and over for you, which is great. That's another wonderful affordance of biology. So that's one piece of it. The other piece that we haven't talked about a lot today has to do with the information storage function of DNA. So DNA is an amazing information storage molecule. It's highly stable. You can copy it pretty well with a very low error rate. You can see I'm trying to think through the exceptions to that, and there's reasons there are exceptions. And as I mentioned before, it's incredibly stable if you keep it dry and cold, and it takes up teeny tiny amounts of space. So there are computing companies that have been working on making DNA, literally using it like computer storage. And so there was a famous collaboration between Microsoft and Twist Biosciences. Twist is one of the leaders in DNA synthesis, where they were literally using it to store computer code. So there was no actual life there. There was no living stuff. It was just using the DNA sequences itself for information storage. And in order to do that, you have to synthesize lots and lots and lots and lots of DNA because the molecules actually are the storage. So lots of really interesting. Another great example I can give you, there's a company in India that was looking to help with food security. And so they were using DNA molecules as a barcode, and they were using that to barcode plants coming from particular farms so that if later there was, say, some sort of salmonella outbreak, you could read off the DNA barcode and track it back to the farm where that actually came from. So that's a really great thing. And then they realized they could build an insurance company around that to help insure the farmers against these possibilities of getting these outbreaks later on. So again, like, we are so fortunate with being able to use biotechnology and look to biotech because biology took care of the building blocks. Now what we're doing is figuring out how to use those building blocks for innovative applications.
That's amazing, and something we'll dive into later if we have time. But just like the fact that that innovation is creating opportunities in the insurance sector, like
Mhm.
the orders of implication, second, third, fourth of this, I think are, not to be cliche, but like only limited by the imagination of like where this stuff can go and where the opportunity lies in any space or industry. So it's super exciting to hear about. As
And
a
the examples we just gave are some really nice examples that lead us to these things. Like you should ask yourself, as a business leader, what should I be doing to take advantage of or to prepare for this bio revolution? Well, the first thing is you've got to have your executives have at least some sort of passing ability to speak the language of biology, or at least to understand it or to get in touch with people who can. That's number one. But number two, you are looking for places where you can harness that language of biology, even if your company isn't a biology company in any sort of traditional sense of the word. And that's where I think communities like Singularity are so great, because we've got this interdisciplinary mindset and this ability, as you said, to look at second, third, fourth order implications of these technologies. And I think that's something we're going to be doing a lot of at the Future Biotech Summit, right?
Yeah, that's exactly the purpose of that program. And it's to your point, the purpose of Singularity. It's hard to do, but it's really fun to do. And it's, as the years go on, a requirement to stay in business and to be successful, is to hold all of these things in your mind at the same time, and get good at collaborating with people who know a lot about this particular stuff, like yourself, to help identify these opportunities and think outside of the kind of the standard channels of business as we know them, especially as the pace of exponential change gets faster and faster, as we always talk about. Um, Going into another question here, what are your thoughts on mirror life?
Oh, I'm so glad you asked that. Okay, so just a few months ago, a big report was released by actually a very large group of very prominent synthetic biologists, raising some warning flags about mirror life. So what's mirror life? Now, I have to be careful because I'm using one of these fancy gesture controlled AI cameras. And the way I would normally explain this, I'm worried is going to trigger my camera to do something weird. So I'm going to be careful here. Actually, maybe what I'll do, Cody, is ask you to make the motions so I don't freak out my AI camera.
All right,
Okay, so
let's see if I can do it.
why are we talking about mirrors when we talk about biology? The answer is because molecules have what we call a handedness. So, Cody, would you please hold up both your hands? So hold them up, palms facing me. Perfect. So now you can see each of his hands has 5 fingers. They're in approximately the same order, but his hands are mirror images of each other, right? He can't overlay them. If you just now, keeping your palms facing, put your hands like one behind the other, so his fingers don't line up properly, right? In order to get his fingers to line up, Cody has to rotate one of his hands and put them palm to palm, right? This is why a right hand glove can't go on your left hand. Thank you. That was lovely. So that's because there's this handedness to it. And biological molecules also have a handedness. And it turns out that the biology and the chemistry work differently if you have a right-handed molecule versus a left-handed molecule. If you remember the horrible disaster that happened with the drug thalidomide decades ago, where women who took thalidomide when they were pregnant ended up having children with awful, awful birth defects, that ended up being the case because the right-handed molecules and the left-handed molecules were mixed up. And one of those did the thing that the molecule was supposed to do, and the other handed molecule made these terrible birth defects. So that was a terrible example of why we have to pay attention to this stuff. So right now, for reasons that we don't actually understand, all of the amino acids that you use in your body have one-handedness, and actually all of the sugars that you use have the other-handedness. I don't have a good example. I don't have a good reason for why that is. But so the idea of mirror life is, what if I could build a bacterium where, let's say, normally all of the amino acids would be right-handed? What if I make all the amino acids left-handed? And then I do the opposite for the sugars. I change the handedness. So now what I've done is made a whole biological system that is internally consistent, but is the exact opposite of my biology. And people think that this could be good for things like using drugs, for example, like drug molecules that are that, because then it can't interface with my native system. So the idea would be there would be fewer side effects and this kind of thing. Or if people are worried about molecules, getting out and spreading in ecosystems, well, maybe this would be a way to stop that from happening. Those are the positive affordances of Mirror Live. The negative affordances are, gosh, like was there actually a reason why biology chose one mirror and not the other? would these mirror organisms be able to outcompete the natural native organisms? There's just so many unknowns. I actually, if I can plug myself for a second, if you guys come over to my Substack, which is called Be Voracious, I wrote a whole article about mirror life when this report came out, and you can check it out there. Cody, maybe I'll just shoot you a link to that if you want to post it somewhere.
Yeah, we'll throw that in
But
the chat and we'll
yeah,
we'll put it in the email afterwards as well.
yeah. So again, like what the working group finally ended was saying was like, look, if we want to look at individual molecules for individual purposes, fine, that makes sense, and we'll have a lot of containment and safety and all of the things that we usually do as science. But this idea of building a whole organism, which by the way, we can't do right now, that's a little beyond our technological capabilities right now. It'd be very hard to do. It's not impossible, it's just really hard. Maybe we need to do a lot more thinking before the technology gets to that point, so we can make sure that we have the safeguards and these various things in place before it actually becomes possible to make mirror life. So the headline for me is stay tuned.
We are getting a ton of questions around ethics in the chat, which I know that you do a lot of thinking and work around. And so I wanted to try to address as many of these as we can at kind of a broad level. And so I'll just run through some of the kind of the generally the questions we're getting. One is around, going from eliminating diseases to into designer babies and managing the ethical considerations of that. One is on the release of CL1 from Cortical, and the advances happening in biological computing, good or bad, interesting. We have a biosecurity researcher, Kath, interested in the general view of potential risk and mitigation strategies as these advances happen, or like where do the guardrails live? And then a couple other just like generally like, yeah, like, what are the ethics of all of this? So with all of that in mind, I want to kind of go into maybe a chunk here where you can speak on the ethics. And I know I think we're gonna do an bioethics workshop at the Future Biotech program as well. But yeah, I want to just
That's
let you
right.
address that.
So just with those four questions, we'll go ahead and spend the next 6 hours together. Is that what I'm hearing?
Yeah, we'll just
Yeah.
keep it running.
We'll just keep going. Okay, so are designer babies ethical for the future of humanity? So that is a very interesting question. So I was recently giving doing a great session with our Singularity friends from South Africa, and the first thing that I got asked was, "Well, when we have designer babies, dot dot dot." And I was like, "Well, wait, you're assuming that we're going to go that direction, so let's hold off on that for a second." So there is a future, I think, in which we decide that having designer babies, meaning like we've done some sort of change to their genetics beforehand, that people decide this is unethical. However, I can also very clearly see a future in which 50 years from now, my son, who is 13, looks me in the eyes and says, "You were holding tools that could have prevented human suffering, and you were afraid to use them." And then I have to explain what that was like for us. So when we think about making these types of changes, we can think about them in two big buckets. We can think about them as therapies, or we can think about them as enhancements. Those are the two big buckets. I don't think a lot of people argue about the therapeutic piece. If you can prevent someone from getting cancer, if you can prevent them from getting sickle cell anemia, if you can prevent them from getting Huntington's or Parkinson's, why wouldn't you do that? Especially if that's an opt-in system, right? With babies, it's harder because the baby is not giving any consent. Now, I would like to point out that I was conceived and born through the traditional system, and I didn't give my consent to anything that I got. So I don't quite understand in a lot of ways that consent piece because there's no consent in biology as it is, right? Like we sort of get what we get. So that's an interesting question there. But then when we talk about enhancements, that I think becomes a much more slippery slope, right? And so people have asked me, like, "Hey, Tiffany, do you know someone who can tweak my embryo so that my kid is a little bigger, a little faster, a little stronger, a little smarter?" So the smart piece is one that people always ask about. And I've heard a rumor that there's at least one company that's doing that now. Um, the problem with that is that intelligence doesn't live on one place on the DNA that we can point to, right? And we also know that there are things you can do already that will make it more likely that your child will get a college education or have a higher paying job. It's things like waiting until you've finished your college degree before having children, or waiting until you've had a PhD. So it's not like we don't have any other options for a lot of these enhancements that we're talking about, particularly with intelligence and good health. So that becomes interesting. Now, I'm going to do something that I hope all of you guys do when you think about these problems. I'm going to argue the opposite now. So, would you ever withhold eyeglasses from your child who couldn't see, right? And you can look at that in a lot of ways. You could say, well, all the other kids in my kid's class are wearing glasses. If he's going to be able to understand the material or compete for a spot in college, he's got to have glasses too. So then, of course, everyone would have glasses, right? Or we have the glasses already. Why would you withhold them? So I can also see a future in which the idea of withholding this technology from your future generations would be highly questionable, either because everybody else is doing it or because, my gosh, why would you hold them back from this particular affordance? And if we stretch that even further, think about space again. So the human body is supposed to be on Earth. It is not supposed to be off Earth, either in microgravity or in places like Mars that have 1/3 gravity. Pretty rotten things happen to the human body, to all living things when you move them. But there are a relatively small number of genes where there are variations in those genes that are already present in people today that you could imagine if we use those for space, it would actually help future, say, Mars inhabitants. And these are things like better bone density, more resistance to radiation, ability to live on less sleep, better memory, ability to function on less oxygen, like all of these things. And so that sort of takes us into a Gattaca world, if you remember that movie, which is still, I think, one of the great biotech movies out there. Maybe not only is it possible to engineer people to be better Mars inhabitants, maybe we also have a moral imperative to not just send people out to suffer and to be sick. Chris Mason thinks that that's actually a moral imperative. So I don't know how all of this is going to work out. All I know is that for me, I would really want it to be an opt-in system rather than an opt-out system. I would love for there to be transparency and reporting and all of these stuff so that we're being very clear about these experiments that we're running, and to go into all of these things with a very humble mindset, thinking, to the best of our knowledge, this is what we understand today. This is what we think is going to happen. And if something else happens, here's how we're going to respond to it. That's my long story about designer babies.
Amazing.
Yeah,
And it's worth noting, I think this is a topic for a completely different conversation, how generational ethics seem to shift as new generations kind of come
yes.
online, and how it seems to advance or shift kind of hand in hand and going in favor with the technology, which is, is that causation or correlation? Who knows? But that's an interesting kind of side note as well.
Hmm.
Um, I don't want to put you on the spot with predictions, but I want to get like a couple of maybe two or three possible futures. So if we're thinking like something we do at Singularity, which is fun, which is sci-fi design, where you use science fiction storytelling to kind of imagine possible futures. We talked about the Avatar world as one. Do you have any kind of like these are some possible futures I could see as a result of synthetic biology and the variety of things we've talked about today?
Sure. So picture a biofabricated world where everyday items are made by synthetic biology, which would be enabling both local and potentially global circular loops or circular ecosystems with our materials. So, imagine having some sort of biological printer sitting on your countertop that can print out not only like the food that you're going to eat that day and your supplements, but will also take that pair of socks that your kid just tore a bunch of holes in, you stick them in, and then out comes some other material that you needed to do something. That is a far off future, but I don't think it's completely impossible as we get better at making these loops of inputs, biological system output, and so on and so forth. And that will also, I think, be a virtuous cycle because the more of those biological materials that we're making, the better inputs that there'll be to another round of that. So I think that's a really interesting, fun future. Another one that I could give you is something like personalized health on demand, where again, you can do really like small batch on demand biological manufacturing tailored to your biology of the active biologics that you need for your health. That's another one. You can tell these two stories I've told you, Cody, are both very decentralized. I think that's another cool thing about synthetic biology. It's been looked at as an obstacle for synbio in the past, like with biofuels. It's really hard to scale these things. You can't just put these cells in a bigger tank and have everything be okay. That's not how biology works. We can keep beating our heads against that problem, and there are good reasons to do that. Or we can say, well, wait, what if we shift to a decentralized mindset with shorter supply chains and you've got everything close by? Might that be a way to both play to biology's strengths and help us with some of these anti-fragility types of strategies that we should be looking at as we move into a more complicated world? So both of those stories I just told you have that theme. The last possible future I'll give you is a climate resilience by design type future. So again, if we accept that these tools are extremely powerful, and we also accept that climate change is an existential threat to humanity and also to our systems, as well as to the rest of the planet, then we've got a chance to be using these tools to not only adapt to climate change, but to mitigate it in the long run, and to set this new future for how we do business as humans that doesn't damage us, doesn't damage the planet, and takes advantage of all of these amazing tools that we have today. I am also really excited about that one. And to me, when I think again about synthetic biology in space, that is climate resilience by design. That is sustainability by design because we're not carrying all these legacy systems with us to Mars. We don't have to do that. And, to make it as simple as possible, there are no petrochemicals on Mars. So there's none of that historical workforce or scalability or capex or any of these things that we've built up over the last 100 years. We get to start over and to say, gosh, if we had known this 100 years ago, would we have made different decisions? I don't know. That's, I think, a question after a couple of glasses of wine that we should talk about. But so those are my possible futures, and you can see they're all positive futures, and
Yes.
those are the futures that I'm working to build.
Amazing. And I think in line with a lot of what we talked about at Singularity, which combines a variety of technologies, are typically very positive because we believe that that is the best way to go create a positive future is to imagine it positively. And then the decentralization thing is really interesting too, because I think we see that kind of through line across a variety of exponential technologies is empowering decentralization, which um, to your point, I think is just more positive in general if you can pull it off. And it seems like in the
Yeah.
past it hasn't been feasible, but as it becomes more feasible, it probably becomes more desirable and interesting and very exciting for human systems in general. Um, we have a question here about sci-fi inspiration.
Mhm.
Battlestar Galactica, Blade Runner.
Mhm.
What are some of your inspirations and are some of these things that, are in the sci-fi movies eventually possible? Um, we'll go quick on this one and then we'll wrap up, but this is kind of just a fun one
Yeah.
to finish on.
So I think right now my favorite bio tackling sci-fi writer is a guy called Blake Crouch. If you watch Dark Matter on what is that on Netflix, whatever that is now, that's him. So he had a book called Upgrade, which I don't want to spoil anything for anyone, but looks at the idea of using biological technologies to essentially force upgrades into humans. And the thing that really caught my eye when I read that book was I had no quibbles with any of the extrapolations he made in terms of the technology. We can't do this stuff today, but there was nothing in his story where I was like, yeah, we can't do that. Like it's
Interesting.
all completely possible. So I highly recommend that one. I like that a lot. I actually think there's a lot of space for more biotech in sci-fi. We tend to see robots and computers and stuff like that and rockets, but we don't tend to see a lot of biology. So that's my favorite one right now.
Awesome. I want to touch quickly on the Future of Biotech program and ask you to kind of share what it is you're going to dive into at that program. But first, I just want to mention, we'll drop a link in the chat. If this topic is interesting to you and you think that it's valuable to your future, to the future of your community, your company or organization or your government, please consider joining us in San Diego in person, or live, virtually on Zoom. We have access to both versions of the program. And just a quick note that we do have, actually, we recently rolled out a scholarship program for U.S. Federal employees who are currently employed or recently unemployed, to join us, complimentary, to continue advancing your work. So take a look at that link to that application is available on the biotech page that's shared in the chat. But yeah, if this is interesting to you and you want to meet Tiffany in person and go deep on some of these things and really hash out the ethics and look at the futures, encourage you to join us. But with that in mind, Tiffany, just real quick at the end here, wanted to ask you what you plan on diving into, because you are one of our curriculum collaborators for this program and have had a big hand in creating what we're going to be going over. And so, what are you excited about for this program?
Uh, well, I'll just talk about my sessions because I'm actually excited about all of the sessions. I'm planning to be there for the whole thing because I just can't wait to hear what all of these people have to say about this stuff. It's so interesting. So, in my sessions, I'll be introducing synthetic biology as a transformative platform technology on par with AI, that's reshaping industries through programmable biology, sustainable materials, and decentralized manufacturing. You guys got a little glimpse of that today. We'll talk a little bit about what SynBio is and what it isn't. And I'm going to take a look at some emerging business models and policy shifts that could be turbocharging this or holding it back. I want to have the chance to talk more about practical steps that business leaders can take to prepare for this bio-enabled future that we're talking about. And through the workshop, and then we're going to be doing the ethics workshop for synthetic biology, which is going to be really great. And I'm so grateful to the Singularity team for collaborating with me on this. But the idea in that session is to inspire all of you to see yourselves as co-creators of a future where biology drives innovation and resilience and sustainability. Even if you're not a biologist, you can be a part of that future. And my session is going to be showing you what that might be like.
Amazing. Thank you. Yeah, super excited for that. It's coming up May 7th through 9th in San Diego. And yeah, I think that's where we'll leave it for today. We're at the top of the hour. There are a ton more questions. This has been a really interesting conversation, and it's just been great to see the demand in the chat and the folks online. So we will send out an email within the next 48 hours with this recording, those links, any useful resources that you should have. And then you could feel free to share this recording with anyone who wasn't able to be here. I just want to thank you, Tiffany, for your time and energy here. And hopefully we can get to do more of these. I think there's a lot of demand around this topic, and particularly now because it's only expanding and only accelerating. So any last thoughts or comments before we close out today?
Well, I'm really looking forward to going through the transcript after this session because you guys have been overwhelming me with how many great questions you have in there. And I think this is so important because when we're talking about a biotech-enabled future, we're talking about the things that we care about, our food, our bodies, our families, our planet, our communities. And so I just think it's great to see all this enthusiasm about where this tech could be taking us. And as members of the Singularity community, you all are positioned to be part of building that future actively. So I hope I'll get to meet you in May in San Diego. Please come say hi if you're there. I'd really love to see you in person. And I think there's lots more conversation to have, and I'm looking forward to doing that with you.
Definitely. Excellent point. The agency is with us. So thanks all for joining us. Have a great day, morning, evening, wherever you are in the world. Thanks for all the engagement, and we'll see you next time. Thanks, Tiffany. I really appreciate it.
Thank you.